Hello, everyone. Welcome back to my recordings of the lectures. And today we're going to review or go over the central dogma of molecular biology and the process of transcription. You are going to be taking this as Cornell notes, so you're going to follow the Cornell note style. Please pause the video and set up your paper at this point. You're going to be setting up pages 27 and 28 to do to basically copy down the information from these notes. Okay, so our essential question for these notes are basically the things that we're going to do by taking these notes is to compare DNA to RNA. And then we're going to go over the process of transcription, which will answer both of our essential questions. Okay, so with this slide, what you need to understand is the central dogma of molecular biology, and the what it, we need to break down that term central dogma. Okay, so first, the central means the main or most important. So central is the main or most important, and dogma is a belief or principle. So that is what central dogma is of molecular biology. So if you're a molecular biologist, this is what you are focused on, or this is your main belief, is that the most important belief in the basis of molecular biology. And that is well, exactly what is that main belief? Well, that main belief or central dogma is that the flow of genetic information in the cells, it goes from DNA to RNA to protein. And this is the central dogma. Focus on this. Make sure you write these things down because this is the basis for molecular biology. And it involves two major steps. The first step is called transcription, and this is the process of making RNA from the DNA. And that is followed by the process of translation, where the protein is made using the RNA. So both of these processes, these two steps, will be broken down. Today's lecture, we're just going to go over transcription. So two main points for this lecture are number one, to compare DNA to RNA, and number two, to fully describe the process of making RNA from the DNA, which is we call transcription. So, to cover the first essential question, what are the differences between DNA and RNA? So, you can either look on page 300 of your book, or you can simply look at these two pictures. Here you've got DNA in the first picture to the left, and you'll see that it's made out of two strands. If you look at the RNA on the right, it's made out of one strand. So there you've got one difference. The second difference, if you look at the list of ingredients on each of the pictures, you've got DNA having thymine, adenine, guanine, and cytosine. If you look at the ingredients on the right for the RNA, you've got adenine, guanine, uracil, and cytosine. You should be able to note a difference between those two. So um, DNA has thymine and RNA has uracil. And finally, the third difference is the kind of sugar. If you look underneath on the pictures, also both pictures, you could see that the sugar in DNA is called deoxyribose, and the sugar in RNA is called ribose. And that is the third difference, the kind of sugar. So, um, what we're going to go over now are the types of RNA found in nature. Okay, so there's three main types of RNAs that we find in cells. The first type is called messenger RNA. In the red arrow on the picture, you could see it's pointing to the mRNA. It looks like one strand. The second type is called ribosomal RNA, and it looks like a glob because it's actually part of the ribosomes. And so you see kind of that kind of blue string running through these. That is the ribosomal DNA. So ribosomal DNA is used in the ribosome. It's part of the ribosome. That's why it's called ribosomal RNA. And then the third type of RNA found in cells are called transfer RNAs, and they kind of look like this T right here at the bottom. Uh, that's a T. That's what tRNA molecules look like. So our RNA has three different shapes. Okay, so here we've got the central dogma, DNA to RNA to protein. And this shows you that the process of transcription is basically taking the DNA and making RNA from the DNA. So that is called transcription. So the first thing we're going to go over is what transcription, the word means. Okay, so the word transcription comes from the word to transcribe. To transcribe is basically to um, uh, transcribe something means to put it in another way, to copy it in another way. In other words, if you've ever had a paper that tells you put it into your own words, like let's say the vocabulary words that you had to do, or boxes, where you had to put it into put it into your own words, basically that is transcription. So they've asked you to transcribe something. And here in the bottom of the page, you have someone that wrote some notes and it's been transcribed into like typing, and it looks different and it's more arranged when they retype it, but basically the both of them say the same thing. So to transcribe is basically to say the same thing but in a different way. So, uh, transcription is kind of like making a copy because you're saying the same thing in a different way. The process by which genetic information on a strand of DNA is used to produce a complementary sequence in RNA. So, in biology, in molecular biology, we basically got the word transcription, to say it in a different way, to describe the process by which DNA is used to make the RNA strand because it's producing a complementary sequence of RNA, and that is what transcription means in molecular biology. And the reason why this has to happen is because the information in the DNA is in the nucleus, and the mechanism to make proteins is outside of the cell. So transcription has to take place in the nucleus where the DNA is. So how does this happen? We're going to go over how to do that in just a moment. So, why do we need to do that? Well, like stated before, DNA cannot leave the nucleus. And like right here in this graphic, help, I can't get out. So the DNA is like stuck inside the nucleus. The ribosome is where the proteins are being made. So the information has to get out to the ribosome, but the DNA is stuck in the nucleus. So how to solve this? Well, a message has to be sent out with the information, and that's where mRNA comes in. mRNA basically takes the information from the DNA, exits the nucleus, and it combines with the ribosome to make proteins. And that is the process of transcription. Okay, so this really is an overview of how this process happens, and we'll go over the details in the following slides. Okay, so now we're going to break it down into transcription into three steps. Step one, this enzyme called DNA polymerase that is there's only in DNA replication that we just covered. There was two enzymes involved in that. That was helicase and DNA polymerase. Those were the parts the enzymes involved in replication or making copies of the DNA. Now what we're doing is that we're making mRNA. How do you make mRNA? Using an enzyme called RNA polymerase. So RNA polymerase unwinds the DNA. So that's step number one. And here in this graphic, you basically saw the two strands separating. And that's what RNA polymerase does, sorry. RNA polymerase just separates a section of the DNA, not the big, it doesn't start from the beginning, it starts later on. It starts at a section that needs to be read. Okay, so once the RNA polymerase separates these two strands, um, RNA polymerase starts adding the same enzyme, starts adding nucleotides to make the mRNA in the 5' to 3' direction. And you will see here in this graphic that it only transcribes one side. It doesn't do both sides, just one side gets transcribed. So you will see here that C attaches, G attaches, the DNA says G, well, the RNA polymerase puts down a C. The DNA says C, the RNA polymerase puts G. The DNA says T, the RNA polymerase puts A. The DNA says C next, the RNA polymerase puts G, and so on. The one difference between you see there that when it's the DNA says A, the mRNA, the RNA polymerase puts U instead of the T because there is no thymine in RNA. It cannot put a T, it puts a U instead. And this allows it to leave the nucleus. So you will see the exception to the base pairing rule is U instead of a T. And RNA polymerase is the enzyme that does all of this. The third and final step is that the DNA returns back to its original state. So it does not stay separated, it goes back to what it was. And the mRNA now, the RNA polymerase goes away, does more, starts creating more mRNA somewhere else, but mRNA polymerase base RNA polymerase just goes away, and the mRNA that's now created leaves the nucleus to the cytoplasm to join up with the ribosome, and the DNA just goes back to being zipped up again. So there is absolutely no changes done to the DNA. The mRNA, even if the mRNA is wrong, it doesn't really matter because the changes are not being done to the permanent structure of the DNA, only to this little section of mRNA, and that one is leaving the nucleus and going to be read. It may make a junk protein, but it doesn't really matter because your body can destroy those junk proteins. It doesn't, even if the mRNA is wrong or was transcribed wrong, it doesn't really matter. Because as long as the DNA is correct, you're good. You can make another mRNA that's right. So the body will do this. Even if you're making junk proteins, it will, mRNAs, because the mRNA is wrong, it doesn't matter. It's only the DNA that matters. Okay, so those were the three steps of transcription. Okay, so in a nutshell, what does all this mean? Complementary mRNA strand. Why is it complementary? Because DNA is the original master plan. It's a template. RNA serves as a blueprint or copy. Precise copying of DNA, what happens if the DNA isn't copied perfectly? Well, the DNA stays the same in the nucleus. It's fine. The RNA goes to the ribosomes in the cytoplasm and creates a junk protein, but who cares? Your body can destroy those. It's not a big deal, as long as there's other pieces of RNA that are made right. Okay, so this is another way of putting the three steps together. They actually have, each step has a name. The first step is called initiation. That's when the RNA polymerase binds to the DNA and separates or unwinds the DNA strand. The second step that we looked at was called elongation. It actually has a name. That's where the RNA polymerase uses the strand of DNA as a template to add nucleotides and to make the mRNA from the 5' to 3' direction, and that's called elongation. That's when the mRNA starts getting longer. And then you've got the termination, that's the end. That's the end of transcription. The transcription stops when the DNA returns back to a double helix, and then the new mRNA created through transcription leaves the nucleus. So that is officially the end of transcription. Okay, here what you're going to do is you're going to copy the black strand and then try to figure out how to transcribe the bottom. So you're basically going to be acting as the RNA polymerase to create the complementary strand in the 5' to 3' direction. But you're going to be, every time you see an A, you're going to add a U instead of a T. And every time you see a T, you're going to add an A. And every time you see a C, you're going to add a G. And every time you see a G, you are going to add a C. So you're going to create these and practice because you're going to have more practice to do in some worksheets. And this is officially the end of this presentation. Once again, I want to remind you of the central dogma of molecular biology. It's that important. And it's basically that DNA gets transcribed to make RNA, and RNA is translated to make proteins. The subject of our next conversation is going to be the second part of protein synthesis, translation. So hope to see you soon. Hope you were able to pause, rewind, and answer all the questions correctly. Have a great day.